Welcome back to the playlist on nucleotide catabolism, and more specifically, this particular playlist is on purine catabolism. Okay, so I want to do a quick recap on what we've been talking about in the past few videos. So, first of all, this video is on urate oxidase, okay, and we'll get into some really important implications on this enzyme later on in the video. But I want to do a quick recap. So, we started off with the adenine component of purine catabolism, and basically what we started off with is this molecule, which was adenosine monophosphate. And then we used a phosphatase to cleave off the 5' phosphate, and we got adenosine. Then we used adenosine deaminase, and that got us to inosine. And then we used an enzyme called purine nucleoside phosphorylase, and that got us to hypoxanthine. And then we did 2 cycles of xanthine oxidase. The first cycle got us xanthine. And then we used the second cycle of xanthine oxidase, or xanthine dehydrogenase, and that got us to urate. And this is sort of where we're picking up right here, is with urate oxidase. Now, there's 2 things that I want to talk about before we get into the heavy-duty mechanics of this enzyme. Number 1, this video on urate oxidase and urate itself as a molecule is going to be dealt with in multiple videos. So if you don't get what you want in this video, there's going to be at least one other video where we go into the heavy-duty mechanics of this enzyme, and we go into all the physiology, organic chemistry, and biochemistry of urate, or sometimes it's referred to as uric acid. Okay, so it's going to be multiple videos. The second thing I want to drive home, which might not be apparent at first, is that this enzyme, which arguably is one of my favorite enzymes to learn about, urate oxidase, is not expressed in humans. Okay, so all this business right here, AMP to adenosine, to inosine, to hypoxanthine, to xanthine, to urate, this is all done in humans. In fact, it's done in pretty much every organism imaginable that we really care about, okay? And most organisms are able to express urate oxidase. They're able to have this enzyme present, which catalyzes the conversion of urate to this molecule right here, which is called hydroxyisourate. Okay, so most organisms can do that. But what we'll find in this video and the next one is that humans lack urate oxidase. Okay, they don't have the enzyme. Now, one thing I want to say about this is that notice I said humans lack the enzyme. Okay? But remember, there is of course a gene that codes for the enzyme, right? Well, it turns out that humans have the gene. We have a urate oxidase gene. The problem is that we don't have the enzyme. So what that means for us is that the gene has somehow, throughout our history, become transcriptionally inactive. So the gene for urate oxidase is there. It's known to exist, but it doesn't get transcribed into mRNA, and therefore if there's no mRNA, there's no protein. Okay, so what that means for humans is that urate is the end product of purine catabolism in humans. Okay, that's very important to understand. Urate is the end product of purine catabolism in humans. Now, other organisms that are not upper-level primates, that are not great apes, that are not hominids, they have the enzyme urate oxidase, and perhaps they have others after that that can make further conversions to other degradation products. But upper-level primates like chimpanzees, gorillas, humans included, upper-level primates, hominids, they all lack this enzyme. And so their degradation product, their final one, is urate, or sometimes referred to as uric acid. Okay? In the next video, we'll delve into more about, the exact nature of what might be a benefit of actually not having this enzyme and some important implications in that. Okay, but I just wanted to get the point across that we have the gene for urate oxidase, but it's not expressed. So humans and other great apes have lost the ability to generate the protein / enzyme urate oxidase. So what ends up happening is if you consume a lot of purines, okay, whenever you consume a purine, the purine could, of course, get incorporated into DNA or RNA. I think that's pretty obvious at this point if you're watching this video. But if, say, you get excess purines, well, you're going to do the catabolic pathway for purines. And by the way, it's not just AMP that gets degraded to urate. It's also GMP, and IMP, and XMP. All of the purines end up getting degraded to urate. Okay? So one important implication that we'll go in more detail in the next video is that whenever you eat purines and they get degraded to urate, the urate will not stay inside the cell. It'll actually go into the blood. And so momentarily what you'll end up with is something called hyperuricemia. Okay? The uric or urus in that name implies that we're talking about uric acid or urate. So hyperuricemia is basically a condition in which we have elevated urate in the blood. Now, in a healthy individual, the urate gets excreted by the kidneys. In fact, one of the proteins that you should be aware of is there's a protein called SLC2A9. This is a protein in the nephron that allows you to have tubular secretion of urate. So in a healthy individual, this is just one of the proteins in the nephron that allows the excretion of urate. So that hyperuricemia should only be temporary. But see, there are conditions where maybe you have damage to these proteins, perhaps. Or things like high fructose corn syrup, inflammatory foods, can actually decrease your tubular secretion of urate. And so this hyperuricemia will be chronically elevated. Okay? And so you'll have trouble excreting the urate. And when urate is not able to be excreted, we'll find in the next video that it's able to crystallize. It forms polymers with other urate molecules and urate-like molecules. And when they form polymers, they crystallize and come out of solution. In other words, the solution is the blood and the lymph, and they come out of it. And these crystals of urate and urate-like molecules, they crystallize, and since those crystals are more dense than water, they tend to sink to some of the lowest possible places in the body, which happen to be things like the big toe. And this process by which the urate and urate-like molecules crystallize and sink down due to gravity, this accumulation of uric acid crystals is called gout. And we'll talk more about this in the next video. We'll talk about the biochemistry and the organic chemistry of this disease. But gout, just to give you some foreshadowing, is an astronomically painful disease. So let me circle it in this bold red color to denote that it's bad. Okay? So the point here that I want to drive at is that humans, like other hominid species and great apes and things like that, we don't have this enzyme. But see, other animals do. So they're able to take this urate and make a conversion to something called hydroxyisourate. And let's actually look at the mechanism of how that occurs. Okay? So here, right here, this is the form of urate that's going to start the mechanism. Okay, this is how we start the mechanism. Now, there are several things to realize about this mechanism in the active site. There's three really critical residues that are involved in Bronsted-Lowry acid-base catalysis. Okay? That's a critical threonine residue, a critical lysine residue, and a critical histidine residue. There's also a water that's held pretty tightly in the active site. So here's what happens. Water initially is going to deprotonate this histidine. That's going to cause the formation of a new Schiff base, whereas the other Schiff base comes out and abstracts a proton from the lysine. And then the lysine will then deprotonate the threonine. And then the threonine will deprotonate this particular nitrogen of the urate molecule, and that's going to cause the formation of an enoloid intermediate. So it's basically a deprotonated hydroxyl group adjacent to a Schiff base. Okay, so that's this functional group right here. It's sort of an enoloid type functional group. Okay. The next step of what's going to happen is, let me scroll over so you can see it. We're basically going to collapse this enoloid here on the other side. So there is a second enoloid that's over here. And once again, we're going to collapse that. So this particular oxygen is going to collapse back down to form the carbonyl. We're going to reform the amide. These pi electrons are going to shift around. And what we're going to do is we're going to initiate an electron transfer. And specifically, the electron transfer is going to happen. I'll draw fishhook arrows to denote that it's a one electron transfer. One of these electrons is going to attack a molecular oxygen in the active site. We'll talk more about that in just a minute. The other one of these electrons, which I'll do in a different color, let me do it in blue, is going to end up on this carbon right there. And notice once again, I did fishhook arrows to denote that these are one electron transfers. So you should expect there to be radical electrons. Okay? Now, when I nucleophilically attack this molecular oxygen with a radical electron, that's going to break one of these bonds, and it's going to come out and abstract a proton from a hydronium that's held pretty tightly in the active site. Okay, so what that generates is this peroxy species right here. This is called a peroxy species, and I generate a purine radical. Notice that I have these two radical electrons right here. Okay. So now what's going to happen is I'm going to get a coupling reaction, okay, just as you would imagine. So these two radical electrons are going to couple. So this is going to come right here. I'm drawing fishhook arrows to denote that they're one electron transfers. And so now what I have is I have this peroxide linkage right here that's now attached to the purine nucleus, okay? The next step of what's going to happen is I'm going to get an elimination. Think back to your organic chemistry. So what's going to happen now is I'm going to get an elimination. So basically what's going to happen is a concerted process in which, I already used green, so let me go back to yellow. So this bond right here, this bond, is going to come out and abstract this proton, and that's going to force these electrons in here to form the Schiff base. Okay? And so what that eliminates at this point is hydrogen peroxide. So hydrogen peroxide is my leaving group. Okay, very rare that we see things like that, but urate oxidase does that. It eliminates hydrogen peroxide. Okay? So basically what happens is the two atoms right here of molecular oxygen both get incorporated into the final hydrogen peroxide molecule. Okay? And that's actually where the term oxidase comes from. If you think back to your definition of an oxidase enzyme, it's an enzyme that uses molecular oxygen, but none of the atoms of molecular oxygen get incorporated into the final product. And that's certainly the case here. They both get lost as hydrogen peroxide. The next step of what's going to happen is called a Schiff base addition reaction. Okay, so basically what's going to happen is, in sort of a concerted process, the water molecule is going to attack this carbon right here, forcing these pi electrons to come out and abstract this proton from the water molecule. And now what you have is you effectively have one form of hydroxyisourate. But see, the mechanism of the enzyme is not yet complete. So what has to happen is this carbonyl has to reform, and that's going to cause a pi electron rearrangement. So these electrons are going to kick in here, and then these pi electrons are going to come off of the Schiff base, and they're going to deprotonate the threonine, which deprotonates the lysine, which deprotonates the histidine, and then, of course, these pi electrons are going to rearrange. And then this nitrogen comes out and reabstracts the proton from a hydronium that's allowed into the active site. And so basically what that means for this enzyme is you get the final product, which is called hydroxyisourate. And hydroxyisourate can react with an enzyme called hydroxyisourate hydrolase. And that product, which we'll look at in other videos, can be transformed into other things that could be excreted by other organisms. But the key takeaway from this video is that urate oxidase, the gene for it, is present in humans, right? But it's transcriptionally inactive. Transcriptionally inactive, meaning that RNA polymerase does not make mRNA from this gene. So it's inactive, and so that means there's no enzyme made, and that leaves uric acid or urate as the final degradation product of purines in humans, upper level primates, great apes, hominids, etc. Okay, so hopefully this video gave you a little bit of intuition on urate oxidase. In the next video, we're going to go over the physiology of some really important things in this, like SLC2A9 and things like it, hyperuricemia, gout. We're going to do some analysis on the structure of urate, see how it might crystallize, things like that. And then we'll also look at perhaps how diet plays into the production of gout and kidney stones and things like that. See you in the next video.